Hi, everybody. Welcome. There's no fun music as you're coming in. I considered it, but right now I'm doing all the tech. So we're just gonna welcome people in. And I'm Katie Kandarian Morris. I'm the minister at the Unitarian Universalist Fellowship of Durango. I want to welcome everyone here and maybe suggest that you set your view to speaker view tonight. And also invite you to mute your computer if it's not already muted, because so we don't have background noise. And I will also go through and check to make sure that people are muted. We were waiting just a little bit because one of our moderators, one of the leaders of this meeting tonight, he is here, good. Okay, he's having a hard time getting in. So now we should all be here in just a minute. Anyway, I want to welcome you to this event tonight and welcome you to the Zoom room. Hi, just a second. We'll go ahead and introduce you in just a minute.
Okay, I apologize for, I don't know what.
That's okay. Just a second. We'll be right with you. So anyway, as I said, I'm Katie. I'm the minister. I welcome you and welcome to the Unitarian Universalist Fellowship of Durango. And I want to introduce our interim director of faith formation, who is Sarah Sauter, and she is going to tell you a little bit about our program this evening.
Thank you very much, Reverend Katie. I'm Sarah Sauter, the Interim Director of Lifespan Faith Formation at the Unitarian Universalist Fellowship of Durango. Tonight's presentation is part of our adult faith formation offerings, and I'm glad you're all here to hear the science behind coronavirus and the vaccine. And I would like to now introduce Jack Turner. Jack developed the CoronaDoc program through the San Juan Basin Health Program, and he administrates it. Jack, can you say hello and introduce our panelists?
Yeah, hi there, everybody. Thank you for inviting us. And the origin of this program, it was really practical in nature. My wife is a general manager at the Strader, and like so many businesses, she had all this, regulations just constantly changing, and there, and there's loans and all this other stuff, and it's been quite an ordeal, this pandemic. And one of the things she had a challenge with in dealing with her employees wasn't just, doing the regulations. If you said, hey, you need to wear your mask, well, if someone was skeptic about wearing masks, the reply would be, well, how do you know? You're not a doctor. And that really occurred to us that during this last year, so many people have never actually talked to a doctor. They get their information about coronavirus off the internet or Facebook or a friend or a neighbor. And so I called up my friend Joe Murphy, who is a doctor in town, and we grew up together here in Durango, and asked him about the idea. Anyway, we got the ball rolling. And so one thing I will do during the conversation is I will type in the address for coronavirus and for the CoronaDoc information. So if any of you know another group that would like to have a presentation, we'd be happy to give it. But I'm just the grunt in this whole thing, and Dr. Goodpastor and Dr. Murphy are the ones who really have so much good information to share with you. They're just wonderful people. And so with that, I'll just say thank you, and thank you for having us too. Take it away. Sarah, you want to go first?
I'll let Joe start. He has the nice intro. But hi, everybody. Nice to see you on Zoom. Thanks for coming. You all know who I am. Most of you do. I'm, mom of Benji and Ellie, and UUFD member and local internist. And let me let Joe go.
Okay, thanks, Sarah, and thank you very much for having us. This has been a very fun project and an interesting project. The way we approach it is we'd like to just give you just a short, maybe 15-minute presentation on the basic science behind viral illness and specifically this viral pandemic. And then we can kind of segue into specifically ways to control it, and specifically vaccines. Um, as Jack said, I was born and raised here in Durango. I practiced here for 36 years, and I actually retired a year and a half ago, as opposed to Sarah, who is in active full-time practice, and amazes me that she has the time to put into this with full-time practice, but I appreciate that. So coronavirus, we've gotten used, in fact, probably most of you know more about viral illness now than you ever wanted to know over the past year. You hear a couple of synonyms for this: COVID-19, which stands for coronavirus disease 2019, when it first was discovered, December of 19, or SARS-CoV-2, which is severe acute respiratory distress syndrome coronavirus 2. And that name actually came from the SARS epidemic, very similar coronavirus that caused an epidemic in 2003 in China, but fortunately was nowhere near as contagious as COVID-19. So those are all the synonyms for this coronavirus. What viruses are is they are very simple organisms. They are genetic material, which is DNA or RNA, that's surrounded by a protein coat. And that's about all that there is to them. That's much different than bacteria like staph, strep, E. coli, which are much larger, more complex cells. There are hundreds of coronaviruses. Most infect only pigs, animals, bats, cats, and other animals. But there are some coronaviruses that cause a common cold in humans. COVID-19 is called a novel coronavirus because it's brand new to our knowledge when it was first discovered back about a year ago. So how do they work? How do viruses cause illness? A virus cannot replicate on their own. If a virus does not get into the cell of a host, of an animal or a human, it just dies, and it does not replicate or form other viruses. But if a virus does get into a living person's cell or a living animal cell, it hijacks their genetic material and basically makes multiple copies of itself, usually probably at least 1,000 copies. So you get this one virus that goes into a human cell, and then it just cranks out more and more copies of its DNA or RNA, coats them with a protein, expels them, and the cell dies. And then you have 1,000 more viruses. The coronavirus is called that because it has this crown of spikes, of protein spikes. And it is those protein spikes that actually allow it to enter a human cell. Those spikes have an affinity for a receptor on the cell called the ACE receptor. And when it is able to fuse with that receptor, it is able to enter the cell and then cause infection. So how's it spread? Everybody's pretty much aware of this, but you may have seen that this evolved with time. When this first came out here in the United States in January, February, and into March, the thought was that this was transmitted by large respiratory droplets. And those large droplets would, if you cough, sneeze, or breathe, would go out several feet, fall down onto whatever you were nearby. So if you recall, there was an emphasis on washing all surfaces, washing your hands, keeping your hands away from your face. All good advice, but it's panned out that really the main way of transmission is small respiratory droplets, small enough to be actually what they call aerosol. And aerosol droplets are suspended in the air for maybe 15, 20 minutes, for quite some time after somebody has coughed, sneezed, or breathed. This is a similar way of transmission to other things that you may be more familiar with, like the common cold, influenza, measles, chickenpox, mumps, and even some bacteria like tuberculosis and diphtheria and whooping cough are all transmitted by respiratory droplets. So COVID-19 disease. This is still a little bit of a puzzle in terms of why everybody doesn't get sick, and especially why 50% of people don't get sick at all. They have no symptoms, even though they will have the virus in their respiratory tract, and it'll be coming out when they cough, breathe, or sneeze. We do know about the risk factors that you may be familiar with. If you have hypertension, you're overweight, if you have diabetes, or if you're older, especially over age 65 to 70, then you're at much more risk for being symptomatic and actually much more risk of dying. Overall mortality is probably 1 to 2%, as opposed to 0.13% for influenza. But if you're over 80, the overall mortality, if you get COVID-19, is 8.4%. And so this is, and the other thing is that the significant contagiousness of it is what has led to the crisis with our hospitals and our ICUs with the pandemic. Fortunately, not that many people get sick, but when you have millions and millions of people that have gotten the virus, it's going to be a stress on the ICU and the system. So the big thing is prevention. How can we prevent this? I won't talk a lot about treatment. Sarah's a little more familiar with that, being closer to current practice. But I think prevention is the important thing. And with any respiratory illness, prevention is avoidance. If you can avoid being where the virus is, you're not going to get sick. Now, that's easy to say, and it's certainly easier if you're like me and you're retired and you can isolate away from people. So isolation, that works, but not particularly practical. And that's why the next two things have become so important. One is social distancing, and they've chosen 6 feet just as basically there's been some studies and experiments that have shown that most respiratory droplets probably drop out at 6 feet or less, even though there is concern if you're in an indoor space or if there's some weak air conditioning and those viruses droplets could spread around. But 6 feet has been thought to be a reasonable distancing. And then masking. Initially, it was a little puzzling, the opposition to masks that occurred. Some of this started clear back in February when the CDC did not recommend masking for probably 2 reasons. One, that they were concerned about the availability of masks to caregivers. But secondly, there was still it wasn't really known that this was transmitted by aerosol. However, by April 3rd, by April 3rd, the CDC, WHO, everybody had come on board and said, yes, masks are important. And all of the data since then has just been more and more supportive of this. If you look at all the studies around the world, populations that mask fairly consistently have a 70% reduction in the amount of disease, the number of cases. So masking definitely works. Now, that brings us to vaccines, which has, of course, been the biggest deal. When I started giving this presentation about 2.5, 3 months ago, no vaccines had been approved yet for use. Fortunately, now we have 2 that have emergency use authorization from the FDA, and a third, I believe Johnson & Johnson has applied. And in fact, they're supposed to go through that process February 26th. So I think maybe I'll hand that over to Sarah, if she can do a little more detailed description of these vaccines and what our anticipation is for the rest of the pandemic.
Thanks, Joe. That's awesome. Well, it's always great to start with Dr. Murphy lays out the biology for us, and so that helps me transition into the vaccines. I am, been involved with science and watching, and learning about scientific stuff for a while. But even I was, wondering about these new vaccines and what's this technology and why is everyone saying it's so new, and is that really safe? And I'm very comfortable with the technology of vaccines. and what they provide. But I curious at the beginning too, what's this mRNA vaccine? So I had to dive in myself, and I do feel like it's easier with a scientific background to become more comfortable with things, because you can relate to other things that and learn. And so I very much appreciate that this new technology can be make us want to say, hey, what is this, or am I sure about this, or let me watch it. But and doing the research, I became more comfortable around this technology, understanding that it had been in development for more than 40 years. There's actually a really amazing story of a female scientist who was instrumental in the development, when people basically told her she was nuts and she really had to push for this. But that began 40 years ago. So one of the things that happened about 20 years ago with this technology that was really revolutionary was they were able to put this into a lipid or fatty coating that allowed the cell to let it enter into the cell so that it could stimulate what it needed to do. They've since been developing this mRNA technology and been using it in HIV vaccine development, as well as anti-cancer drugs. So this has been under development for some time heavily. The way I think it's helpful to kind of go through the way the vaccine works, the mRNA vaccine, that's the one that we have currently two, the Moderna and the Pfizer. So I think it's helpful to kind of go through the mechanism. And it's really ingenious what they did. So we've all seen the picture of the coronavirus molecule, and we see the spikes that are coming out of it. What they've done is they were able to sequence the genetic code just for the spike. So it's not for the entire COVID virus, which is, as Dr. Murphy said, an RNA virus. And RNA is just a code. We have the DNA, which is the double-stranded. The RNA is just a single strand. All RNA does in our body is code for proteins that we make every single day. So we have tons of messenger RNA running around in our cells to code for every protein that makes us work. And basically when we do make a protein, it then the cell spits it out and the mRNA degrades, dies, goes away, it's gone, cleared by the body. So they were able to get the genetic code for just that spike protein and essentially sequence it. And then they were able to code it in this lipid or fatty coating so that it could go into the cell. So what happens is you've got that that little bit of that mRNA and it's in solution, and you get the vaccine. And then basically we go to biology, the class that we, freshman biology that we all had to sit through, right? So everybody remembers the cell. You've got the cytoplasm inside the cell. You've got the nucleus sitting in one small area, which contains DNA. And then you have all the cellular machinery that makes the cell do jobs, right? So if you remember the mitochondria, the powerhouse of the cell, you may or may not remember the ribosomes. So the ribosomes are the portion of the cell that makes these proteins. And essentially what I like to think of, and you can see, is like, if this is the mRNA on a little bit of code, it goes into the ribosomes, comes out the other side, what you get is the protein. So it's just like this manufacturing little part of the cell. And so the mRNA never goes near the nucleus. It never goes near DNA. It only is a code that codes for proteins. So you get that mRNA for that spike protein, it goes through, comes out, and your cell starts spitting out spike protein. Same concept as when you get infected with the virus. That virus is just spitting in its own mRNA, which is for the whole thing, and it starts spitting it out. And off you go. And so all the virus is replicating. In this case, it's just that spike protein. So that goes out of the cell, and it's essentially, then your immune system is on alert, and it says invader, invader, and it activates all the things it needs to do to go after an invader. And it's essentially learning. It's learning about this thing. It's training all these different parts of the immune system to recognize this single spike protein. And so you have a B cell side of our immune system and a T cell side of our immune system. You often think of antibodies, that's more on the B cell side, but we do have these killer T cells, which for viruses are one of the main portions of the immune system that take out viruses. So that gets activated by both disease and vaccine. Then the mRNA that was there is just going to degrade like mRNA normally does. So that's going to be gone. But what's going to be hanging out is the spike protein, and your body's going to be going after it with all these little things. So it's going to make antibodies and do all this. They're going to glom onto that spike protein and clear it like they normally do with everything else. It makes this complex and it gets cleared. So, but our immune system is amazing, and that it has these things called, it makes memory, it has memory B cells, we make antibodies and all this. And so you are essentially training your immune system with the vaccine. And so then, the spike protein gets degraded, it gets taken out, and that's gone. And it's like, okay, we're back the same. So we have like a semi-trained immune system, so that when it sees the real COVID molecule with all these spike proteins, all these spike proteins on the outside, it's going to be like, whoa, got this. I know what this is, and grab onto it and take it out and get rid of it before it has any opportunity to go bananas. Because what it loves to do is go in, hijack the virus. When it comes in, it loves to go in, hijack, and just replicate. And it doesn't care about anything. It just wants to replicate. Well, in the meantime, it's destroying cells that it's in and causing massive inflammation. It's the virus is getting in your heart, your brain, your lungs, all your tissues, and massive replication. So the idea is, and why some people do better and others, do worse, is essentially you've got your immune system, and how well can it shut down a virus from replicating and not allow it to get out of control. So essentially people who have conditions that suppress their immune system, and we know that, we've talked about excess weight being that, diabetes, people who are on immunosuppressive drugs, people who possibly even low vitamin D has been linked to a weakened immune function. If you don't get on that virus right away, those are the people that it replicates in and it gets out of control. And none of us knows. I mean, we can postulate who's a higher risk person, but there have been people that have been 25 and 18 and, have gotten an overwhelming case, and we don't know why that happens with them. But the key is that your immune system's trained. So after the first dose of these two vaccines, we know that we have some limited protection. I think, gosh, I'm already blanking on this. I can't believe it, but it's 64% with the Pfizer, and I think it's 80% with the Moderna. And then after both doses, within a week to 14 days, you have your highest level of efficacy, which is between 95 and 100% against severe disease that would cause hospitalization and death. So the way they designed these trials was, well, I'm going too far. So anyway, that's what happens with the vaccine. You can consider it like an immune system training, and it's only trained against the spike protein. And they do think that even with variants, what they're seeing now is that there's enough similarity that the vaccines are still going to be trained well enough. We might need a booster to increase the efficacy, but the reality is that we never expected to get 95% efficacy out of a vaccine. I mean, people were literally blown away when this happened, because for the seasonal flu, it may be 40 to 65% efficacy, depending on the match for that year. So it's a really remarkable scientific achievement that it was able to match this well. When they designed the trials, they were not testing everybody every week just to see what would happen. They basically, they had a periodic, a pre-test before the second dose that was a routine thing, but otherwise it was requiring symptoms to be tested. So that's why we can't say that there was no asymptomatic infection. We can say that there was no symptomatic infection that then resulted in hospitalization and death. So for instance, with the Pfizer trial, I think it was the Moderna, actually the Moderna trial, there were 30 cases in the placebo that got a severe infection requiring hospitalization, and there were 0 in the vaccine. Subsequently, they did identify one case that ended up being positive in the test and was hospitalized. That person was post-vaccine number 1. So the second vaccine is important to get the full efficacy, because, right, I said that if you're with Pfizer, you've got 65% efficacy. So you do want to complete the series. But, you know, the vaccine side effects, some people ask me about that a lot, you know, those are typical immune reactions. So when you get the flu, the feeling of the flu is because of the immune system going after the influenza virus. So, so that's the same thing. When you have a highly efficacious vaccine like this one, or if anybody's had Shingrix, which is also 90%, it is stimulating your immune system, you know, and so all those effects are the ones that people talk about: headache, fatigue, body aches, maybe a fever. The strongest symptoms that are most common are headache and fatigue, and a local arm reaction. So, but those were the biggest things. And, you know, you can feel comfortable that that's your immune system activating. And then don't worry if it's not, because a lot of people get worried when it's not, when you don't get symptoms. But the idea is that, you know, you're training your immune system with the two doses. The Pfizer and Moderna vaccines, they were just studied this way. So they just are different doses of the exact same thing, essentially. They both got a hold of the code, genetic code. It's a shared information once it was sequenced. Pfizer decided to do 30 micrograms in their doses. Moderna decided to do 100. So you are getting roughly 3 times the dose when you get a Moderna vaccine. So some people have anecdotally reported stronger reaction. We see this sometimes too with things like the flu vaccine and the high dose flu vaccine. So we see different doses for things. Sometimes the other thing I wanted to just bring up was that there is some real world post-vaccination study data right now being collected. So Pfizer has a partnership in Israel where they basically have vaccinated 90% of people over age 60. So they have 300,000 people in this trial right now. And basically they've seen their case rate drop dramatically in their 60 plus age range. And the interesting thing that they're finding also is that people have 4 times less viral load in their nose after vaccination. So why does this matter? This is talking about the carrier status or the asymptomatic spreader. So it's really one of the questions we're trying to ask, right, is, well, am I going to spread this or could I get this? And that's been the most recent thing is, well, you still could possibly spread, you know, even though you're not yourself going to get a severe case of COVID after being vaccinated, you could give a mild case or an asymptomatic case to an unvaccinated person. So what's great to see in this trial is that the viral load in the nares is low. And I was also listening to a great presenter today from UCSF who was saying why she thinks we're going to see good data showing that the spread is low after vaccination based on the amount of immunoglobulin or antibodies that they're collecting in the nose after vaccination also is a good sign that, again, we're going to have just an active level of protection so that carrier status will be lower. That kind of brings us to like, why would we want to continue to wear masks and things? And basically, what they're recommending now is essentially kind of a tiered system of protection. So they call it be sure to message with optimism about the vaccine. And I think it is something to be optimistic. And they said, you know, if you're looking at this tiered system, things that would be acceptable would be things like, if 2 people are vaccinated, could they interact together and feel safe? And the answer is yes. 2 vaccinated people could interact together and feel safe. Could a vaccinated and an unvaccinated person interact without masks on and all the precautions? Not at this time. So there's still that risk to that unvaccinated person because we don't have the clear data to show us that that we're not going to spread a case to them. And then, of course, an unvaccinated to unvaccinated, we still are very high risk situation there. The other thing that they've talked about, which I think is really interesting, is kind of a triangular approach to prevention. So Dr. Murphy talked about the social distancing. And then there's another aspect of that, which is the masks. And then the third portion of the triangle is the ventilation, which he brought up. And kind of the way you can think about it is, I think, intuitive as to what you've been doing, right? If you're in a well-ventilated space, like going on a hike together and you're staying apart, you might be choosing to take your mask off. But if you're in a poorly ventilated place, then you're going to wear your mask and you're going to try to social distance. And if you're in a place where you can't social distance at all, you really need to make sure that you've got your mask on and possibly even now a double mask. And so these are the kind of decisions you sort of have to make to reduce your risk. If you want to be really low risk, which a lot of people have done, is they're making sure they only see people in well-ventilated areas with their mask on and they stay 6 feet apart when they go hiking. I mean, that's the ultimate protection, right? When they talk about masks now, they've got some great information here now, basically saying what kind of masks should we be doing? We should really be thinking at this point, if we're outside, a simple cloth or a surgical mask is acceptable. If you're indoors, that's where we want to really think about fit and filtration is what they're talking about. So the best would be an N95 because it has the best fit and filtration. The next would be a surgical mask with the cloth lying over it, the cloth mask over it. And that's because of the fit and the ability to get a little bit better. The surgical mask has these fibrous things that really help with the not allowing those small droplet particles through. And then you've got an extra cloth one that's going to help you make your seal. The other that they said, is acceptable as well as doing the cloth mask that fits well with a filter. So that's going to be your best bet, and that's going to be the continued recommendation for masking in order to keep this, keep yourself protected while you're still doing this. Why would we want to still mask? Why would we want to still take all these precautions? You guys have all heard about the variants coming out. The issue with viruses, especially whether it's DNA or RNA, is they are not smart. They just want to replicate. They're just these pieces of genetic material. They just want to make themselves over and over again. They are not very accurate. So when they replicate, and the more they replicate, the more they're going to make mistakes. Some of these mistakes will be meaningless and we won't even hear about them. There's certainly thousands of what we call escape mutations that have occurred with this virus, but they're not meaningful. And the ones that we're hearing about are the meaningful ones that are more contagious. They're being able to make themselves, spread faster. And so we're hearing about the British and the South African and the Brazilian, and now there's a California. The best thing we can do is try to reduce transmission, and we do that through all that triangle approach that we've had, and by vaccinating, so that we don't, there is, like I said, good evidence that the viral load's reduced in the nares, and there'll be less transmission. So if we can do that, then we can stop the mutations from occurring and potentially, stop a mutation that's really detrimental to either more transmissibility or more virulent, where it could cause more death. So that's why we really want to keep this down. I think we've done an amazing job in the Plata County with our masking and our social distancing and everything we've done. And we've really been able to keep this at a reasonable, it got scary there in January with ICU capacity, but it's been obviously much better than our neighbors in Farmington and what they've suffered with. So I congratulate our community on what we've done. And I think that diving into the vaccine is just our next greatest hope to where we can get back to normal, supporting our businesses, our people, our jobs, our families. And so I'm really, really excited that we have this opportunity for vaccination. The ways to get vaccinated that I know of right now, if you're a patient of Mercy and you're at the eligible level, you'll be invited through your portal. They are getting limited supplies, so it's always a lottery system. The same is for San Juan Basin. They're handling all the vaccine and distributing it through other channels. One of them is the community COVID vaccination team that takes place at the fairgrounds. You sign up through that on the San Juan Basin Public Health website where it says Community COVID Vaccination Interest Form. Just make sure your name's on there. If you're in the eligible category, they're pulling all those. Again, randomized lottery. We did 300 initially, and I think last week we did 620, and the goal is to be doing 1,000 Mercy's now looking at that site to do during the week. So that will be even more vaccines that we can get out. Ash is doing some, Access Health, City Market, Albertsons. I hear new ones every day. So you guys probably know more than me, to be honest. But that's my general thing that I like to be sure to tell everybody about those things. And I hope that's helpful in explaining a little bit behind the science. The J&J vaccine, the new Johnson & Johnson vaccine, is essentially, it's going to be the same thing, except for that portion of the mRNA is going to be inserted into what's called an adenovirus, which is responsible for very common illness, very infectious, so they basically took out a portion of that viral code and stuck the mRNA in the adenovirus, and they're going to insert it that way. That was just a vector for the mRNA, because there was also always this concern about, will the cell accept this? And just that was their vector they chose to do. And they've had good outcomes too. I was looking at the J&J data today because, as Dr. Murphy said, they are looking to approve it on an emergency basis on February 26th. So that trial had 43,000 participants. It was a lot more diverse. It was in the US, Central America, and South Africa. More people of color were represented, more people with comorbidities, and even more people above 65 years old. So it was a pretty amazing situation. And turns out it is protecting against the South African variant pretty well. It's not at 100% like it is with the others. And like the other vaccines are 95 to 100%, but it was, even with the variants in South Africa, 85% protective against severe disease. So that's impressive as well. Yeah, I'll stop there and we can open it for questions.
Thank you so much, Sarah, and thank you so much, Dr. Joe. If you wouldn't mind, if you have questions for either one of the docs, if you could just type them into the chat line, and I'll draw your attention to the post that says, while we welcome all your questions, we can't answer specific questions to your personal medical conditions. You'll need to contact your personal physician for that. So we do have one question in here from Pedro asking about, since the more contagious strains are beginning to circulate, and some that the vaccine may or may not prevent, do you think there's any hope that choirs could start singing together again? Who would like to give that one a shot? Dr. Sarah, Dr. Joe?
I guess I could go ahead and comment on that. I think there's still optimism that choirs are going to sing again. And it's for some of the reasons that Sarah, Dr. Goodpastor mentioned. Even though these more resistant variants, or excuse me, more contagious variants are circulating, and certainly they're going to be here and they're going to be more, the vaccines still have been shown to be overall effective against those. And I think all of the measures that we are doing now, I really believe that all the measures we are doing now are going to eventually result in at least some component of herd immunity. Now, will that be late this summer, next fall, sometime next winter? Maybe so, but just because of the remarkable success of the vaccines and the fact that they still have at least some effectiveness against those mutant variants, I'm optimistic for choir folks. Just can't say what the time frame would be. Dr. Goodpastor, do you have more to add to that?
I would agree with you. The one thing I saw that was really interesting that I haven't had a chance to read, but it was on this UCSF talk today. There's an article apparently in the Scientific American called "Vaccines Not Need Completely Stop COVID Transmission to Curb the Pandemic." So I'm interested in reading that, and I'll post that if I can find that in the chat, because what the ID doctor said was just really interesting, was that there's a way that we can curb the pandemic it doesn't have to, we can do it in many mechanisms. Basically, if we get enough people vaccinated and we get enough people, doing the right things, we can really curb the pandemic. So it kind of relates to the variants question. What if we don't get the same efficacy that we were getting despite the variants. They have not had time, obviously, to do studies in people with the new variant and protection against that, but they have done Petri dish studies, and they've put the neutralizing antibodies that that are developed to the vaccine onto the variants, and they have shown good efficacy with the neutralizing antibodies in vitro. So that is, yes. And so that is a positive thing, that the neutralizing antibodies are present. That being said, the vaccine companies are already working on boosters related to this. So there's a question in the chat about, might we need a booster? And that's a very real possibility that we need a booster each year. And Moderna's already announced that they're working on that too. So, it could end up being like seasonal flu. And I think, that's unfortunate, but it's a global world, and we're interconnected, and we've had a new virus come on, and it was probably expected that this would happen. When we saw the first two, SARS and MERS, the only reason they didn't get to this level is because they were so deadly. They killed their hosts. So the issue with this thing is that, like everybody's talked about, you can spread it around everywhere, and some people aren't going to get sick. And so it's, we were looking at, what did we say? 78% of the deaths have been in people 70 and above. So, but that there's all these other folks out there, they're able to spread it. So I was looking at allergic reactions. Oh, somebody asked if any of the shots being given are placebos. In the trial, yes, there was definitely a placebo arm. So that's how they were able to tell. So in each of the trials, I'd say probably more than 100,000 people have been studied so far, if you look at the trials, because Pfizer was about 30,000. Moderna, I have the numbers here. Moderna was 30,351. J&J was 43,783. Pfizer was 43,448, and they would have split that down the middle for placebo versus vaccine. And like I said, for the Pfizer, they had 170 COVID infections that were severe enough, and they were, at all. And the placebo group had 162, and in the vaccine group, there was no severe disease. In the Moderna, it was 196 symptomatic infections were identified, 30 in the placebo group, 0 initially in the vaccinated group. But then I did mention they did find one PCR positive person who was hospitalized, but that person had only received 1 dose of the vaccine. In terms of severe allergic reactions, the contraindication to the vaccine right now is a severe allergic reaction to a component of the COVID-19 vaccine. The biggest suspect is PEG, which is like a pegylated, mixed thing that's a component of the vaccine. And that would be a severe anaphylactic reaction to that. Some people do know that. We are being cautious with people who have had reaction or allergic, like anaphylactic reactions to other vaccines or shellfish or bees or peanut butter or anything like that. It's still recommended to receive the vaccine. And even my husband received it. He has a shellfish allergy and has had anaphylaxis. But basically what we do is we watch them longer. So everybody gets watched for 15 minutes because anaphylaxis is an immediate reaction and should appear within 15 minutes. We watch people who have a history of that for a longer period of time for 30 minutes. So every place that you receive vaccine should have the capability to treat anaphylaxis. So like we're doing it at the fairgrounds, we have EMS there, we have a crash cart inside the building, but we also have full EMS there, and it should be that way anywhere you get it. I recommend to my patients that tell me they have a history of anaphylaxis, I've been recommending they try to get it in a facility that's well equipped, to make sure, if it's at the fire department or where is it, or if you're going to get it at the grocery store, ask them, what would they do if you had anaphylaxis? They need to have an answer. We have an EpiPen, we have this. The good news is anaphylaxis is treatable. They've had it in a rare number of patients, and it's a treatable condition. It's not caused death.
And there's now enough data on that the risk of anaphylaxis with these vaccines is between 2 and 6 patients per million vaccine doses. So again, very, very rare. And that's why it's being recommended for everybody unless they've had a specific anaphylaxis to an ingredient of that vaccine.
Great. Thank you. There's also a question in the chat line about if one can still get the virus or carry the virus once vaccinated, how do the vaccines promote herd immunity?
So,
Well,
so
I think,
oh,
go ahead, Sarah. Go ahead.
that's the data that I was mentioning. So that's when we're talking about people who could be what we call carriers, right? They're asymptomatic themselves. They have no symptoms, but they could carry, or maybe they have mild disease and they could spread it to somebody and thinking that, oh, I just have a sore throat or something. I have allergies or something like that. So the trials weren't designed to find out about asymptomatic disease because they did not just systematically test people every single week to see if they were COVID positive. They waited for people to have symptoms and then they tested them. So that's the data we have right now. But there are ongoing trials testing people all the time. I think the AstraZeneca is one. But what we do know, and the reason we think we're going to have good information and about the fact that there's less virus replicating in our nares is because they have been able to show in this real life trial in Israel with 300,000 participants so far that the replication of the virus in our nose, which is where basically we're giving it to people, right? It's in our nasopharynx. That's why the first test was right up in there, and it still is. If you get a COVID test, you get it here, because that's the virus heavily replicates there, and it's basically a proxy for what's going on in the body. So the fact that they're already showing that the viral load replication after vaccination is 4 times lower. That gives us a great feeling knowing that we are going to be able to prove that soon. And then also, as I mentioned, one of the infectious disease doctors talked about why she believes we're going to prove soon that it decreases transmission. And that's because they actually measured antibodies inside the nasal mucosa, and they are very high. They have very high antibodies against the spike protein. So that's a positive sign, probably the reason the viral load's very low. We know that systemic vaccines like this that we get induce the entire mucosa to develop what's called IgA antibodies. Those are specific antibodies that are just in the linings of our stomachs, the mucosal linings of our nose, mouth, stomach, intestines, all that. That specific antibody has been induced by this. So that's a positive. So we'll be coated, from the top all the way down with the antibodies. They also did some early studies in macaws, before they even went to human trials. And they were able to show that they could block viral replication in the lower airways and the upper airways. So they basically took swabs of the lower airways and the lungs through a special procedure in these macaws. And they took nasal swabs, and basically they had no or very low viral RNA found after vaccination. So based on this science that we have now, we think we're going to have data soon, and we think we're going in the direction that's going to prove that we're not going to be big carriers and we're not going to be big transmitters. But nobody can tell you that for sure right now. Does that make sense?
Sure.
All that scientific story. But does that make sense, what we know now and what we're hoping to find out soon?
Yep.
Yeah, I think this just comes into what you would call an abundance of caution, because every virologist is looking at all the evidence that Dr. Goodpastor cited and saying, there should be very little chance that there is going to be transmission from vaccinated people. We just don't have that 100% hard and fast proof yet, but I think we're getting close. There was an interesting quote, and I watched the same video that Dr. Goodpastor did. There's a quote of a virologist in back east who said, the thought that there is going to be transmission after vaccination is the ultimate pessimism. Nobody believes it's going to happen, but you just got to be cautious until they can prove it.
There's also, Dr. Murphy, a question in the chat about the efficacy, and the person is asking for some clarification of that. The number 95%, does that mean this 95% that you won't get the virus, or that you, even if you're asymptomatic, or that you won't get sick if you get the
Well,
virus?
the data for the 95% number is getting sick with symptoms, because when they did the trials for Pfizer and Moderna, that's what they looked at. And it's actually a little confusing, but it made sense with the numbers that Dr. Goodpastor just mentioned. On the Pfizer vaccine, 162 people in the placebo arm got sick with COVID. 8 in the vaccine arm got sick. That 8, basically that represents 5%. So there's only it's 95% successful at keeping you from getting sick. There was just that 5%, that 8 compared to 168. The 8 is 5% of 168. So maybe that makes a little more sense that way.
And the 100% is against getting severe illness. And
Right.
you know, what that same person said on that was great, which is, listen, if we all get the sniffles, or we all get, you know, a little cold, great. We can live with that, right? What we can't live with is the risk of somebody dying because of this, which, you know, I'm hearing of people every day. I keep thinking, I can't believe we're still doing this. But I had somebody just tell me that their 63-year-old airline pilot died still not wearing a mask when he went to get his haircut, and the barber called 3 days later. So, you know, an airline pilot's probably pretty healthy. They have to pass a test, you know, and it's just so sad to me that even as recent as 2 weeks ago, we're still losing people to basic precautions, you know. But, you know, I really believe the vaccine, if we can just get this vaccine out to as many people as possible and just get our immune systems trained against this novel virus, then, you know, we don't have to worry about that risk of people dying. It's the main thing. Not necessarily just getting sick, but dying.
And what about the length of efficacy? There's a question in there about that. What do we know?
At the moment, we can only follow the people who have been in the trials. So we've got about 6 to 8 months worth of data, and they're going to continue to follow them. So we're just going to have to keep following them. One of the folks said that was really great is that we know for like the influenza that there are these things called memory B cells, and they tested a woman who was in her 90s, and she still had memory B cells that protected her from the influenza that occurred as a child. So there's the big buzzword is apparently the Tcell response. So you know, we all talk about antibody response and whatnot, and so they follow antibodies, but the reality is that the antibodies can wane, but we still have this entire other arm of our immune system that's the Tcell, and that appears to be the most robust and probably the longest lasting. So they are testing that in labs, but it's not like something that would be available to any of us to test our Tcell activity against this. But that's why we can't just say, okay, the antibodies are gone, now I'm in trouble, or antibodies are here, I'm good. That Tcell system is still functioning. So we'll have to follow those trial participants.
Great, thank you. And folks, I'll invite your final questions in the chat line, but here's one more about the preservative in the multi-dose vials. Is there mercury in that? And are we worried about that?
No, there is no mercury in these multi-dose vials. They're like Dr. Goodpastor said, this PEG is a preservative that apparently there's been a concern about very, very rare anaphylactic reactions to that, but the vials are mercury free. Do you know any other information on that, Sarah?
No, I mean, it's very rare anymore for them to use thimerosal as a preservative, but they certainly aren't using it in this this particular, they're not using mercury. That'd be a no-go for so many people. They're just kind of beyond using that anymore because that's such a no-go in vaccines anymore. So
Great, good to hear. Doesn't appear to be other questions in the chat line, so I'd like to take this time to thank you, Dr. Sarah Goodpastor, and thank you, Dr. Joe Murphy, for coming to us this evening and helping set us straight on the vaccinations and the disease itself. Katie, did you have any final words of wisdom to say to us before we adjourn?
Yes, because I didn't do any at the beginning. You might have noticed if you're a regular comer to UUFD that I had a chalice already lit here, just with all the good thoughts and hopes. But as we go, words from Kendall Gibbons that says, there is finally only one thing required of us, that is to take life whole, the sunlight and shadows together, to live the life that is given us with courage and humor and truth. So it's another reason we value truth, and we're so glad that these physicians could be with us to offer this good science and their experience. So thank you.
May I say one last thing?
You bet, Jack. You bet.
Yeah, I just wanted you to know that Dr. Murphy and Dr. Goodpastor have been doing these presentations over and over and over, all volunteer, and they've just got the same warm bedside manner, Zoom side manner. And I can't begin to tell how much I admire them. And they're also volunteers down at the vaccine clinics we're doing at the fairgrounds. So it's quite a community we have here. And thank you for inviting us to be here tonight.
Thank you, Jack, for making this happen. We're
To
grateful.
everyone, and if
I didn't start the pandemic.
Thank
Blame
goodness.
me.
And Dr. Sarah, Dr. Joe, Reverend Katie, and Jack, if you could stay with us for a photo that's been requested, that would be great. And the rest of you, we bid you a great evening. Have a great evening. Take care.
thanks for
Thank
having
Good
us.
night, good evening.